again. Oh, there we go. Andrew just did it. Perfect.
Sorry,
Okay.
I was doing it at the same time.
Yeah, no, that's great. Perfect.
All right, so we want to have a break now, but we'll have one, say, in 25, 30 minutes, okay? And if we don't finish today, we'll carry on tomorrow as well, all right, with this section as well,
Yeah,
in the middle.
we like finished day one just now.
I know.
We have an hour
Finished
left.
day one and day two, yeah. That was tough to get the timings right.
But I mean,
all right, so
I really appreciate all the questions that people
oh
have been
yeah,
asking.
yeah, no, this is a lot more fun. This is a lot more fun.
It's more fun and it's more useful
Yeah.
to you than just us droning on about stuff that you don't understand.
So what I'm going to talk about is bistability. Okay, so I'm just going to share screen. There it is. Bistability. So what is bistability? So I got a definition there. A bistable system has two stable states, and it can switch between them. Okay, I'll show you some examples in a minute, but biological examples include differentiation. So when a stem cell turns into, say, a liver cell, that's some kind of bistable transition. It's a very special one, though, because it's irreversible. My liver cells don't spontaneously transition back to a stem cell, and they don't spontaneously transition into nerve cells, for example. So cells have a special way of locking in transitions in bistable systems. But anyway, so that's what a bistable system is. Another one, of course, the most famous one, perhaps, is the lac operon, where E. coli switches from glucose to lactose. That's a bistable system. It's either consuming glucose or consuming lactose. And then there are other ones like in the MAP kinase pathway and signaling networks. You find some evidence for bistability there. Now, one of the key features of bistability is positive feedback. And positive feedback is one of the primary ways of making systems bistable. And I've got a sort of schematic here on the right of a gene, which is producing a protein X at a rate V1. And this protein feeds back to itself to stimulate its own production. V2 is just the degradation step for the protein. So this is a sort of a very simple motif showing positive feedback, and therefore this system will show bistability. Now you may be thinking, yeah, but do they exist? Well, they do. They're actually more common than you may think. There was a famous researcher at Caltech, Davidson, who worked for many years on elucidating the gene regulatory network for sea urchins, the developmental circuitry that was involved in development of sea urchins. And this is just one image from one of their papers. You'll see this all over the internet. If you look up sea urchin gene regulatory network, you'll probably find a bunch of these pictures. Some of them are really complex, but if you start looking at these, you'll see various motifs. And I've circled one down here. Whatever this gene is, OTX, it's got a positive feedback. In fact, it's got two positive feedbacks because you can trace the blue line all the way back to another gene called BLIMP1, which is positively regulated, and then BLIMP1 positively regulates OTX again. In fact, this is doubly positively fed, two positive feedbacks. The other thing you'll see, of course, are negative feedbacks. They're pretty common as well. Here, FOXA, you have a negative feedback going to FOXA. Another FOXA up here. So you'll see positive and negative feedback in these gene regulatory networks. I really like these diagrams. A lot of people don't draw them like this. I wish they would, because they really give you the sense of a circuit diagram in these gene networks. A lot of the time, you'll just see these hairballs. I'm sure you probably know what I mean. I mean, if I look up one, gene network hairball or something. There you go, that kind of thing, which I think are completely worthless. You really can't make head nor tail of those. But on the other hand, if you look up gene network, Davidson sea urchin not urchin, sea urchin, you'll see these kinds of things, right? And I think these are much better at things at looking at, and these, here we go. Oops, where did I go away? Ah, God. If I zoom in on that, so you see that's a bit dense, that one, but you'll see circuits like this, and these give you a lot more information than those hairballs. In fact, we're about to start a project on another grant I've got to try to generate the E. coli gene regulatory network in this form, because at the moment people just publish them as hairballs, and I really don't see the point of that at all. So we're going to try and generate one of these for E. coli. So give us an idea of what the circuit looks like. Anyway, back to the slides. Got a view. Oops, okay. There you go. Anyway, so you do find genes with positive feedback. You'll find positive feedback sometimes in metabolic networks. You'll find them in signaling networks, and you certainly find them in gene regulatory networks. So I got some pictures here that tries to depict what do we mean by a stable state. So I mentioned here, back here, stable states. What do we actually mean by that? And there are, if you go online, you'll see various pictures of people trying to, trying to show you what that means. You can think of a dynamical system as a landscape, and that landscape has bumps and hills over it, and the canyons represent the steady state. And these canyons, once the system goes into this canyon, it finds it very hard to get out of. Okay, so we refer to these as stable states. All right. And this is an example of a stable state, all right? You're going through a transition, and you settle out to some stable state. What's happened is the system has found itself in one of these canyons, and it's sort of stuck in there, all right? Okay, and here's a really simple example. This is a really simple model. X0 goes to S1, S1 goes to who knows. And I do a run of simulation, and I get this curve, okay? Now I can tell that it's stable because I can add an event to this model. And here I'm just upregulating S1 a little bit. And as Lucien said, this is a one-time transition. So when the transition occurs, and it'll only happen once, right? Because time only transitions above 10 once. When that happens, it executes S1 equals S1 times 2. And because the transition only happens once, S1 equals S1 times 2 only happens once. So that's why I see this spike happening. So S1 gets sort of doubled in concentration. After that, nothing else happens to the event. But now then we see the decay back to the original level of S1. All right. So this one we can be sure this is a stable, steady state. Okay. Now, what's the sort of picture one can imagine if you had more than one stable state? Well, if you have more than one stable state, let's say you had two, that must mean there are two valleys in your landscape or two canyons in your landscape. It also implies that to get from one canyon to the other, you have to go over a hill. And of course, that hill is not a very stable place to be. You can either roll down one side or the other side. Now, you could balance yourself exactly on the hill if you're very careful, and you can stay there, but a slight push and you'll go one way or the other. And so this picture also depicts the idea of an unstable state. Okay, so bistable states are often of this kind. They'll have two stable states, two canyons, and then a hill in between that represents the unstable state. Okay, and we'll do this in a simulation. I'll go back to Tellurium. I'll show this, but you can imagine a system like this will have maybe a high state and a low state of some kind, right? But the concentrations between these two, the concentrations at these two canyons will be different. They won't be the same, all right? And you often find that there's a high state where the concentrations are high and a low state where the concentrations are low. They're both sitting in their little canyon. But in between these two, there's probably an unstable state. Okay. Oops. All right. So let me just check here quickly. Yeah. I want to show you something just to convince you of something. If I go to Google, let me go to Google and I type in light switch toggle, you see things like this. Good old Amazon. Okay, that's a common light switch you might find on your wall. It might be slightly different depending on what country you come from. But that switch obviously has an on state and an off state. And those two states represent two stable states. All right? Because I can touch that switch and it won't easily switch to on or off. I really have to push it in order to make it go on or off. So the switch is quite stable. On the other hand, I can move that switch so that it's in the middle, and if you're really careful, you can make it sit in literally in the middle, and it's neither off nor on. But that is an unstable state because I only have to touch it a little bit, and the switch will either flip to the off or the on, because there's a bunch of springs in there that are holding it. And so this is a sort of a common example of a bistable system. Okay. So oops, let's go back to this. All right. So how are we going to build a bistable switch? So to build a bistable switch, we need some kind of positive feedback. All right. And this is the simplest possible model you can make to show bistability. All right. And what I'm going to do is I'm going to drag, oops, hold on. I'm going to grab this. I can get to them. I'm going to grab this, okay, and then we'll go over this, all right, in Tellurium. So by the way, if I'm in a Google Drive like this, and anybody has this, this is not special to us. If you right click and look down here, you see a doc, a sheet, a slide, a form. If you go more, you'll see Google Colab. And it'll create a Google Colab for me, all right? And there it is. So now I can do pip install Tellurium, was obviously forgotten. Then run that.
Not as TE, just pip install Tellurium.
Oh, what am I doing? Yeah,
Dash
sorry.
Q if you want to quiet.
I've really confused it now, haven't I? Could not find. Yes, I know. There you go. Okay, so once it's loaded, right, getting ready.
You can paste in your code in the new thing.
I don't know if I
while
can.
you
Can I?
wait.
Oh, I can. Yeah. Okay, let's not forget to do the restart. And then import, thank you. Okay, it's new code, and then I'll paste. Okay, so this is the model I had on the slide. So let's go through this. I'm importing Tellurium. I don't really need to do that. I just did it, but anyway, it doesn't matter if you import again. I need matplotlib because I'm going to do a legend. But here's the model. Simple model, just two steps. I have x0 going to S1. X0 is fixed. I have S1 just draining out. S1 draining out is a really simple first order, nothing special there. But this first reaction is quite different. And it has two parts to it. It has this bit, x0, and the second part plus this. So it's sum of two rates. The first rate is fixed, and that rate equals the level of x0, which is down here 0.5. This bit here, though, adds an extra rate on top. But that rate is a function of S1. And this equation is written in such a way that as S1 increases, this increases. And it increases quite rapidly because I've got a to the power of n, right? This will give me a sigmoid response to the increase in the rate. So once a little bit of S1 is made, this rate will be fairly low, but it'll start making more S1 as a result. But once S1 reaches high enough, this equation reaches that steep sigmoid portion, and you'll see a rapid rise in the rate, and thereby a rapid rise in S1. So that's the model, and that's the positive feedback. So what I'm going to do now is I'm going to run a simulation at different starting values for S1. All right. So I'm going to start this simulation at different levels of S1 from 0.5 to 3. I'm going to assign S1 to S1 in the model. Then I'm going to run a simulation. Then I'm going to start this. I'm going to use this plot array to start stacking up the simulations one after the other, okay? I've got myself a label, which will show up in the legend. And I'm going to make sure that we have a new color for each plot. So let's run this and see what happens. Okay, so this is what I get. This is rather strange looking because it looks like I'm getting two very different behaviors. So on some of them, just green to brown, if I set the S1 to these levels, the simulation seems to go to a high value, 5. On the other hand, if the S1 is small enough, it seems to go to a lower one. So these are actually two distinct steady states. And in fact, just to convince you, I'll run this for, say, 40, right? And they stay there, right? Not only that, let me see if I can do this. At time greater than, let's see, should we try, let's transition at 20. Let's bump it up a little bit. Let's bump S1 up a little bit to see what happens, okay? Let's do it a little bit more. There you go. So I'm perturbing each state separately, and you can see that both of them are stable, right? Both of them recover from this perturbation. So these two states are actually quite stable. And this is an example of a bistable system, okay? All right, let me get back to the slides.
Herbert, if you instead of running simulate, if you called the steady state function, would you get basically the same results?
Yeah, I'll show you that shortly.
Okay.
Yeah, that something interesting happens with that. Yeah, I'll definitely show you that. I've got it in the slides here. Okay, so what's causing this? You know, why is it that we have bistability here? Now, if you look at some basic behaviors of enzymes and proteins, they often boil down to just 3. So what I've plotted here is the rate versus, you know, some input like a substrate concentration or a ligand on a receptor, membrane receptor. You basically only see largely 3 kinds. You either see a linear response, a hyperbolic response, or a sigmoid response. There aren't that many more. The only other thing I can think of is you sometimes get inhibition at high ligand concentration, and it sort of dips back down, but they're not so common. All right. So we only see bistability when we have a sigmoid response. So it's something to do with this sort of, you know, S-shaped curve. And just to show you what the equations look like, so for a linear, it's just k times x. For a hyperbolic, it's sort of Michaelis-Menten-like, s over k plus s. And then for a sigmoid, it's just a modified Michaelis-Menten with a power on the ligand, right, on s, okay? And that's what I did in the model. And you can increase the sigmoidicity just by increasing n, all right? The higher the n, the sharper the s. And when n is 1, it reverts back to hyperbolic. I don't want to show you that yet. That's the model again. Okay, so that's the result we just got. We had both a high state and a low state. And the question I want to ask is, why does this happen? All right. Oh yeah, so this just explains, this is for your notes. This explains what the code does. That gives me a range of starting values. This does the plotting and so on. Okay, I've gone over that. All right. So why is it bistable? So the way to explain this is to look at each rate individually. And what I'm gonna grab. Oops, hold on. I'm gonna grab. Where am I here? I'm gonna grab this. Okay. And I'm gonna go back to wherever it was. Here it was. And I'll paste that into here. Let me just run it first, make sure it works. Okay. So what I'm doing here is I'm doing this thing I did before in the morning where I'm just plotting the rates. All right. I'm not actually doing a simulation. I'm literally just plotting the rate through J1 and the rate through J2. And I'm plotting them at the same time. Right. I'm changing S1. I'm plotting this, and I'm changing S1, and I'm plotting that. That's all I'm doing. I'm not actually doing a simulation, okay? So down here, I'm going to collect all the information I need. The x-axis is going to be S1. Then the two y-axes are going to be the rate through J1 and the rate through J2. So I've got those ready. Then I start a loop, and I collect all the information, and then I plot it. That's all I'm doing. And this is what I see. Now, the orange one is the easy one. The orange one is just that simple rate law there, and that's linear. The rate is linear in S1. All right, so in fact, that legend is totally wrong. That should be Y label. Let's do that right. Strictly speaking, that should be capital J, but that doesn't really matter. Okay, so here we are. So the x-axis is S1, and the two y-axes are the rates through J1 and J2. J2 is just a linear function, so I get a straight line. The J1, however, is this sigmoid function that's lifted a little bit by the presence of this extra, what I call a basal rate, which is why it doesn't start at 0. It starts a little bit above 0. In fact, it starts at 0.5. Okay, so it starts at 0.5. But you'll notice you get this nice S shape, right? That's the sigmoid. And if you notice, you'll see that these two lines cross at 3 points. Now at these points, let's take the upper one, right? This upper one here. What that means is at this point, both J1 and J2 equal each other. That's when they're equal to each other. And when they're equal to each other, we know that the rate of change of S1 must be 0. And we know that when the rate of change of S1 is 0, we must be at steady state. So this crossing point, if you draw a line down here all the way down to the x-axis, that actually is the steady state level of S1. Of course, there's another crossing point way down here. That's also another steady state. That's at about whatever it is, 0.2 or something. But you notice because of the S-shaped curve, there's actually a middle crossing point. And that middle crossing point actually represents the unstable state. So the two ends represent the two canyons, and the middle represents the hill in between them, okay? All right, let's go back to the slides. So that's what that shows. Now, so that sort of partly explains why we see bistability, but we can go further than that, though. Let me do the slideshow here, okay? So that's the plot I just showed. Now what I can do is I can do a sort of thought experiment here. So X is the S1, okay? And then the two rates J1 and J2. And we're at let's say we're at the upper crossing point. Let's say I make a perturbation, delta X perturbation in X. And I can do that with an event, for example, all right? And if I do that, all the rates change. So V2 changes to this point, and V1 changes to this point. They're no longer equal. That means we're no longer at steady state. But notice that when we do this, the V2 rate is bigger than the V1 rate. Right? V2 is bigger than V1. Right? So these are the 2 points. And what does that mean? If V2 is bigger than V1, that means I'll be consuming X in this case faster than I'll be producing it. And that means, therefore, dx by dx is negative, and therefore X will start to decrease. So if I make a positive perturbation in X, the forces will try to pull the X back, right? They'll try to pull the perturbation back, and it pulls it back to this point. All right, the same thing happens if I were to make a negative perturbation. You notice the lines, the blue and orange lines are flipped. And so if I make a negative perturbation, it'll also get pulled back to this point. All right, so this is why what I meant by it is like a canyon in a way, because if you try to move away from the canyon, the forces will pull you back in. And that's essentially what's happening here. The same argument happens down here. You can do the same thought experiment on the lower one, and you'll find that this one is also an attracting point, right? Everything tries to gets attracted to that. Question is, what happens to the middle one? The middle one is quite different. If I were to make a positive perturbation in X on the middle one, right? That means I move up to here somewhere, let's say. Yeah, these arrows are not very good. But it means that the blue one is bigger than the orange one, and that means V1 is bigger than V2. So that means I'm now making more X than consuming it. And so X will continue to increase. In fact, it'll move from this point all the way to the upper one, which is the stable one. So we can say that this point is unstable. If I make a negative perturbation, the opposite will happen, and it'll get pulled to this bottom one. Okay. Well, that's a bit better. So dx by dt is positive. Therefore, it continues to move it away from this point, or in fact, it goes all the way up to this top point. An example of this, as I said before, is the lac operon. And the positive feedback is from the lactose binding to a transcription factor called LAC-I. There's actually, it's an interesting one because there are two negative effects here, but when two negatives are combined, negative times negative gives you positive, so you end up with a positive effect. And so when lactose is in the environment, it gets transported in, and it activates LAC-I, which then activates the lac operon, which makes more transporter, which brings more lactose in. So that's sort of an example of the positive feedback here. Sometimes it's not so obvious where the positive feedback is, because if you just looked at this, you just see two negatives, and that may not look like a positive feedback. But then you realize what the two negatives combined together effectively form a positive feedback. Okay, oh, that's where that one. I wondered where that picture went. Okay, so that's everyday example of bistability. All right, what else can we do with this? Now let's show you one thing on the on this one. Let me go back to this. What happens if I really make a big change? Because I suggested that you could transition from one to the other, maybe. Although they're stable, if I can make the perturbation big enough, maybe I can get them over that hill and over to the other one. And so let's see if I can, I think we'll look at the bottom one, okay? So I'm going to take out the top ones, all right? And just to show you what that means, I'm going to take out these, there's one more. So I've only got let me just drop it to 1.0, okay? So that means I've only got one simulation now, and it's the low state, okay? And so it's low, it's around about 0.2 or whatever. And I do a perturbation, and it just returns back to that state. I can make a much bigger perturbation. Let's multiply it by 5 and see what happens. Okay, so that's a pretty big perturbation. It's still stable to that one. I obviously haven't managed to get over the hill yet. So let's do 7. Oh, there it is. But let me drop it down a bit. Let's see if I can make it a little bit more. Oh yeah, okay, that's a little bit better. So we're at the lower stable state. This is the spike. And the spike is big enough that I've pushed it over the hill, and it's now in the other stable canyon, and you can see that it transitions up to the high state. And if I can really go to town here, because I can actually switch it back to the low state. If I, let's say, go to 35, where is it, 35, and drop it back down to, let's drop it down, let's go 1 times 0.2. Let's see if I can force it back down. There you go, I forced it back down as well, okay? And this gives you a clue maybe as to how a certain kind of oscillator works. If I could arrange it so that I could flip the system back and forth like this, somehow, I could create an oscillator. And in fact, that there's a one class of oscillator that works exactly like this, but we'll talk about that later. Okay. Now, where am I? Here. Okay, now I'm going to tell you something without giving any justification. That would take too long, but some of you may know this already. How can we identify programmatically, right? Say, without doing a simulation, how can I tell if a steady state is stable or not? There are certain things called eigenvalues, and some of you may have come across these. They relate to the time constants of the system at a given steady state. Now, these time constants are normally negative, meaning that the thing decays. But if you compute these eigenvalues and you find there's a positive time constant among them, it means it's unstable, all right? So I'm just going to give you this as a rule. I'm not going to justify it any further. But what you need to do is you need to compute these things called eigenvalues, which are basically time constants. There's as many eigenvalues as there are variables in your model. So if I've got 2 variables, I'll have 2 eigenvalues. And you basically look at the signs. You just look at them to see if they're negative or positive. If they're negative, it means the system is decaying and therefore it's stable. If one of them, at least one of them, is positive, it means the system is going to expand and therefore it's instability or unstable, okay? Now, in order to do this properly, you have to evaluate them at the steady state. That's not steady state, at the steady state, okay? And that's what we're going to do next. What have I got here? Yes. Okay, let's do some experiments, and we'll bring in those eigenvalues. Okay, so I'm going to grab this model. Okay, I'm going to grab this one. I'm going to put it down in a new cell. Have I a new cell here? Give me a new cell. Oh, here, down here, new cell. All right, there's my model. And I'm going to start a loop. Let's say 10 will do. And what I'm going to do is I'm also going to bring in the random number generator. Now, remember before I was able to spike the system to move it back and forth. Well, what I'm going to do here is I'm going to set the concentration of S1 randomly, some random value, okay? This will give me a number between 0 and 1. I want a number between 0 and 10. I think that should work. So I just multiply by 10. And then I'm going to ask it for the steady state. All right. Then I'm going to print out the value of S1. All right. So let's see what happens. Nothing happens because I made a mistake. Cannot perform. Oh, I've got to know. Yeah, sorry. Event. Yeah, that's sort of mini bug actually. Oh no, it's not a bug. It's not a. Yeah, it's not.
Yeah, it's
Yeah,
because
it's
the
sort of a mini
events.
bug, but it's not a big deal. When you're computing steady states, events don't make too much sense because the event is going to mess up the calculation for that. So generally, if you want to work out the steady state, take out the events and set your system up into the right state for you. Okay. So I've taken out the events here. All right, now let's run it. Okay, let's see what we have. Okay, so look at this. If you can see it, I have two different values for S1. I have a high value of 5, and I have a low value of 1.49. Okay, so that looks like there are two steady states here. Let me do something else. Let me see. Let me bring that down to 5. Change the random number generator a bit. Now let's take it down to 4. And there you go. Right. Okay. Now, if you look carefully now, you'll see there's 3. There's 1.49. There's 5.14. And there's 0.2513. Turns out it looks like there's 3 steady states in this system. So I call this the cheap way of trying to figure out how many states a system has. You just randomize the starting conditions and just compute the steady state, see how many you get, okay? And if you're lucky, you'll find all of them. In this case, I found all 3 of them. The question now, though, is which of these are stable and which of them are unstable? Okay, so let's do this. So I'm going to comment this out, all right? I'm going to grab these 3 numbers. I'm going to put them in there, and I'm going to set S1 to each of them. This is going to be my starting points. So I know these are the 3 states, but I'm only going to look at one at a time. Okay. All right. Now, if I can compute the steady state of this, and I'll confirm that it really is at 5.14. Let me just run this. Okay, yeah, it's 5.14. Right, so that's a steady state. The question is it stable or not? The way to do that is to ask for the eigenvalues. Come on, get full get full eigenvalues. I'm not going to go into how these are calculated or what they really are. Some of you may know that already. But these are basically time constants of the system, and they can tell you more what the system is going to do based on their values. But one of the key thing is if you want to look at stability, you must ask for the steady state. You must compute these at the steady state, okay? So let's run this. Now, to add one other little complication, eigenvalues can be complex numbers. So because of that, you'll get this little, I hope you can read that, you get this little term here, plus 0j. J is the complex number, the imaginary number. Let's ignore that for now, okay? And let's look at what's called the real part. And notice the real part, it's negative. It's minus 1.84. Time constant is negative. That means the system will contract if you perturb it. In other words, it'll return back to the steady state. So I confirmed that the 5.145 is in fact a stable steady state. Let's try another one. Let's try the S1. Do the same thing. Also negative 1.957. This is also stable. Finally, okay, fun bit. Let's try this middle one. And it's positive, 3.00519. Because it's positive, I know this system is unstable, all right? So let's do an experiment. I'm going to grab this model again. And what I'm going to do, create a new code block. I'm going to set the system at the unstable point. And I'm going to run a simulation from this stable point. And I'll run it. A straight line. Now you're thinking, yeah, hold on a minute. You said this was unstable, so why is it not doing anything? Shouldn't it be moving away? Yes, it should be moving away, but if you're exactly on the unstable state, you're basically on the top of that hill, really finely balanced, right? It's like the toggle switch. You're halfway to on and off. You're very delicately balanced between the two. I can disturb it and put it off balance, so to speak. Let's do it at 7.5.
Not if.
And I shall disturb it a little bit.
At,
Okay.
at the beginning there instead of if.
Oops, I think we'll do
Yeah.
at. I'll just really do at. Disturb it a little bit. And then it transitioned. Let me make the disturbance a little smaller. Okay, I'll make it even smaller. It doesn't take much to off balance it, okay? And notice it's transitioning to the upper one. That's because I made the transition above the unstable state. If I make the transition below, like slightly below the unstable one, it should transition down. There goes the transition down, okay? All right, so we really do have three states here. We have a high one, a low one, and a middle one. And the middle one is unstable. All right. Okay. So any questions on that? Are we okay? Oops, where am I? I'm lost. Here we go. All right, let's go on to continue the slideshow then. So this is what we just did. I found it. You might have to do this a number of times and change the range for the random number generation to find them. But it's a quick, cheap way of doing it, because what you can do is you could run this, 10,000 times, and then just write a little script to search for how many different steady states did it find. All right, you can just do brute force finding of the steady states. That's possible.
We do have a question from
Yep.
someone here.
What's that?
Go ahead. I don't know how you pronounce your name. Ayoyon?
What's this? If x equals 0, then despair. No. Uh, what's the question?
Someone just raised their
Oh.
hand. They'll have to unmute and ask it. Maybe they accidentally clicked raise hand.
All right, let me have a look at some of these questions here. Oh yeah, about Sidescape and hairballs. Yeah, agreed. Yeah. By the way, those gene circuits were drawn by a tool called biotapestry, which is still available, I believe. And then there's something about Yuri Alon describes as a long-term memory. Yeah, I'm going to show you something shortly, which is more like long-term memory, because it is long-term memory in the sense that, yeah, if I transition up, it's very difficult for it to transition down unless I impose a really, high disturbance, a really major disturbance. Then I'll flip to the other one. But if they're just normal small disturbances, it'll stay there. And in terms of long-term memory, you could argue that if I'm at the low state, okay, and there was an event in the environment, the system can detect that event, causing the low state to transition to the high state. And in that sense, it's recorded that disturbance in the environment because it's now transitioned to the high state. And that high state will remain there, forever, unless there's a very major disturbance to pull it back down. But a few hours later, right, you can look at that high state and say, ah, yes, that's a record of an event that occurred a few hours ago. So in that sense, it's a long-term memory. All right. Does that answer your question? It's sort of remembering what happened, right? So if I go back to this, I mean, I see what he's saying. This transition here, if I start, let me start at the low state. It'd be better. Let me comment that out, just to show that we really know what's going. So that's the low state, right? Along comes some external event, right? And this external event, say, happens at time 7.5, and it disturbs some signal, which forces the system to go up. Okay. Now, if the cell were to look at the state of S120, at time 20, it would see a high state. And in that sense, it's a record of something that happened at time 7.5. So in that sense, it's a memory, a long-term memory of something that happened much earlier in time. So I think that's what Yuri's referring to, okay? And I'm sure cells do this. I'm sure they do this to record events. So especially during differentiation. But I'll show you something cute shortly about these bistable systems, which might be more interesting. Did we have a hand up? Yes, sorry. You've been waiting.
We had a hand up, but I think it might be a mistake. They haven't said anything.
Oh, okay.
So
Let me see. There's some other
but feel
questions
free
here.
to say anything if you like.
Yeah, it's like balancing a fork on a spoon in a table. Yeah, there are all sorts of little analogies you can come up with. I mean, bistability is really common, ultimately. Yeah, the two steady states are not always identical. Almost always the case. Yeah, almost always non-identical. Do you
Well, I
set?
mean, that question actually was about like the last digits, and some of the things were slightly different from each
Oh,
other, and that's just like
oh, that's numerical error. Yeah,
yeah, just
yeah,
numerical
yeah,
errors. It got close enough, and
yeah. Oh
clearly
yeah, the
it was
random
dumb.
number generator. Very good point. If you want to, yeah, let's say you've done this, this scan of, randomly generating initial points and you've got it, and you want to show somebody else, like in a week's time. If you run it again, you'll get a different answer, but you want to try and reproduce what you did, you need to set the random number generator seed and record that seed. And then you can show somebody next week or next month exactly the same run. That's true. Okay. All right. So looking at eigenvalues. Yeah. Now, what I showed you was, I need to go back to the figure, sorry, was this kind of system. A single positive feedback, linear pathway. That's the simplest possible kind. But that's not the only way to do it. And another common way, and perhaps more common, is to do something called a toggle switch. So I got these pictures from a paper. This is a popular thing with the synthetic biologists, but you also find it in biology as well. It's where you have two positive feedbacks working on each other. It's very common in electronics. So on the left-hand side, we see an electronic circuit. That's often, in the old days, this was a very common way of implementing memory, right? Computer memory. These days, they have other ways of doing it, mainly because this takes up too many transistors. But in biology, you'll find it. This is the lambda phage switch, where it decides whether to go to sleep or just to grow and escape from the E. coli cell. And then this is the synthetic one that was made. Do I have so this is a sort of diagram of a toggle switch. This is based on the synthetic one done by Gardner, Cantor, and Collins back in 2000. This is one of the first synthetic networks ever built. And this basically shows what they did. They had, excuse me, two genes, repressor 1 and repressor 2. Repressor 1 was attached to a reporter, a GFP reporter. In front of each repressor was a promoter, and the repressor was actually a transcription factor for the other promoter, all right? And it was a negative repressor. So repressor 1 negatively repressed repressor 2, and repressor 2 negatively repressed repressor 1. Now, as I said before, two negatives makes a positive, and this is effectively a positive feedback loop, okay? And you can write some simple equations that describes this. And if you do this, you'll discover this system has two stable steady states. And because this group, I don't know if I have it in the next slide or not, but they actually built this in vivo and showed that it had two states. Now, they only gave the equations though. Okay, and this can happen sometimes. Now to model this with Tellurium, you could either literally just enter those differential equations using the prime notation. So I could go U prime equals and V prime equals, or you could try to reverse engineer the network, right? And reconstruct the actual network that this represents. And that's what I've gone and done here, okay? So in order to reconstruct the network, I had to do four reactions. Let me show you the second one and the fourth one first. So the second one is this degradation rate, and the fourth one is this degradation rate. There's a negative in front of them. That's why I know it's a degradation. And I model a degradation just by doing this. U goes to nothing, V goes to nothing. Okay, so those are the two degradation steps. I've got the equations here. The other two steps represent the formation of the corresponding transcription factor. Okay, so I'm making U, and it's a function of V. So remember that U is controlled by V and V is controlled by U. But it has to be done so that V represses U and U represses V, which is why I've only got V in the denominator and I've only got U in the denominator. By putting them in the denominators, V will act as a repressor, not an activator. U will also act as a repressor, not an act as a repressor, not an activator. Right? I could make it an activator just by putting V in the numerator as well. Right, but we want repressors. Remember the picture? It's repressing. Repressor 1 is repressing repressor 2, and repressor 2 is repressing repressor 1. So that's what I have to do here. Then I set up some initial conditions and run a simulation. So let's do that. It's better if we do this live, so you get a better feel for it. So I'll create a code, paste, run. Invalid operator. Oh yeah. Oh, be careful. PowerPoint or slides, wrong kind of quotation marks. Okay, that's not super exciting. Let me start it off. No, I don't want to change that. Let me start it off at 3. And you'll see the transition. There you go. So that's one state. We've got a high state on U and a low state on V. Excuse me. That's a very characteristic of a toggle switch. In a toggle switch, you have 2 variables, not 1. And this, in the steady state, one will be high, the other will be low, and vice versa. If I stick an event in here, I can actually transition it from one to the other. Let me see. Let's make let's see, U equals, let's say U equals 1 and V equals 4. Let's see what will that do. I can force it, there you go. I can force it to flip around, okay? So in the first one, U is high, right? Then I force U down and force U up. And if I do it enough, if I do them high enough and low enough, I can make them completely flip around. So that's a toggle switch, all right? So the difference here with the toggle switch and the other one, this is 2 variables. And they basically complement each other. When one's high, the other's low, and the other's low, the other's high. Whereas the other one, I only had 1 variable, which was S1. It's either high or low. Right? Now, this also has an unstable one, though, in the middle. I don't know. Can I find it? Let me see. Let me see if I can find it. Let's see. Oops. Let's do that. So I'm gonna make yeah, let's just do this and see what happens. To be honest with you, I've never done with a toggle switch. This is gonna be interesting. And then print out U and print out V. Let's see what happens. What have I done wrong? Oops. Oh yeah, get rid of the event. Whenever you're doing steady state, you need to remove the events, okay? Yeah, let's see what we've got down here. Oh yeah, it's flipping nicely. Look at that. So you got low to high, low to high, high to low, high to low. And here's the middle one. That's the unstable one. Notice that the U and V are exactly the same. I wonder if anybody knows why that's the case. Why do you think the unstable state, the U and V are exactly the same? Why do you think that is? Anybody got a clue? Look, it's 1.29991, 1.29991. In fact, even the last few digits are the same. Why is that?
Not only that, but in the other steady states, they are exact mirrors of each other.
Oh, yeah, they're all mirrors of each other. What's
Yeah.
going on here?
I think A1 is equal to A2 and all the degradation and activation rates are equal, I guess.
Exactly. Because it's totally symmetrical, right? So let's make it unsymmetrical. Let's pick something, you know, some weird ones. Let's see what we get now. Yeah, there you go. They're no longer symmetrical. Are they not?
Yeah, you got all three, I think.
They're not symmetrical anymore. Okay, and I got all three. All right. So that was the reason for the symmetry. All right. So okay, so that's nice. Okay, what else have I got here? I may have covered some of this. Yeah, I covered this. You can do the eigenvalues just as before. It'll show you the same information. Okay. Command is get full eigenvalues. Ignore the complex, the O.j, ignore the complex term. Just look at the so-called real parts. All right, and if they're both negative, now we've got two variables, so we have two eigenvalues, right? They're both negative, stable. If one of them is positive, the system's unstable. Okay, and this I show. Here's a transition I show, and there's a little bit of an oscillation there as well as it transitions from one state one to the other. And I can't guess, and I said I can keep on doing that. I can keep transitioning the toggle back and forth all the time. All right, so what else we got? Yeah, okay. I want to show you one last thing, and then we'll actually, we're probably coming to the end, aren't we? Yeah, I'll show one last thing then, right? The last 10 minutes. And then we'll call it a day, okay? And then we'll do oscillators tomorrow then, all right? There's this nice paper some of you may have come across. Sniffers, buzzers, toggles, and blinkers. It's from John Tyson and Catherine Chen and Bella Novak. John Tyson is a famous modeler of cell cycle. And Bella Novak is currently in Oxford. I'm not sure where Catherine Chen is now, but I'm sure she's around somewhere. Anyway, they wrote this very nice paper. It's well worth a read. And they have all sorts of these motifs. I'm only showing you some of them here that have different kinds of behaviors. The one, yeah, you know, they have a whole bunch of them here, and they show all the different behaviors. The one I want to look at is this one, okay? So let's have a look close up of this one. So this one has a signal S that activates this reaction here that produces something called R, and R gets degraded as a result of something called E. Now E can get converted to something called EP via R, and EP can get converted back to E. Now you can think of E and EP as phosphorylated and unphosphorylated protein. And you can think of R as a kinase that phosphorylates E to EP, okay? And E, unphosphorylated form, is the active form that degrades the kinase, okay? And so I had to reverse engineer this because John Tyson gave all the equations as differential equations. And so I reversed those back into, you know, a reaction format, and you can see it wasn't difficult to do. You can see it here. I've got the 4 reactions, 1, 2, 3, 4. The first one has a basal rate K0, and then it's linear in the signal. The degradation of R is a function of the unphosphorylated E protein, but there's 2 components here. It has a basal degradation rate, K2 times R, but it also has this extra degradation rate as a result of E. And then you have this cycle here from phosphorylated to unphosphorylated. And they modeled it using a Michaelis-Menten equation, and that's what I've got here. I have to make sure that R, the kinase, is in the numerator of the Michaelis-Menten equation. But the rest of it is fairly standard Michaelis-Menten. You have the substrate E or EP, and you have the KMs there. And I got his values that he provided. These are the Ks and the KMs and so on. And I got some initial numbers from his paper as well. And that was the model. Okay. So what does it do? Is there positive feedback here? So this one is not, this one is more subtle. Let's imagine I increase S. If I increase S, R will increase. Okay. If R increases, this rate will increase. That rate increases because of the K2 times R, okay? So increases in S will initially cause R to increase and this rate to increase, okay? However, increases in R will also cause phosphorylation of E to EP, and that reduces E. Since it reduces E, the rate here actually goes down. And if the rate goes down, that means R is consumed at a smaller rate. And if R is consumed at a smaller rate, R will increase. And if R increases, it phosphorylates more E. And there's even less E here, and R increases even further. So that's the positive feedback loop, okay? So if S increases, R increases a lot more than you'd expect. All right. So that's the
Just to
positive
confirm
feedback.
for people, the dashed lines in this case mean activation.
Oh,
right?
yeah, right. Yeah.
Okay.
Yeah. That's right. Yeah. Yeah. In other words, S isn't consumed here. E isn't consumed by this, and R isn't consumed by this. Yeah, that's an important point, actually. Well, in this case, it's a very, what's causing activation? Somebody asks. Well, in this case, the model is very simple. We don't know actually how the activation is occurring, but the way it's modeled in the equation is just that there's a second rate component on the first reaction, for example. So there's a basal rate of K0, and then there's this extra bit here that's added on, and that's a function of S. So mathematically, all that's happening is if S goes up, this bit goes up, and therefore it increases the rate, and therefore it acts as an activator. Now, in biological terms, we don't know. It's not been specified what mechanism actually does that, but we could imagine things that are happening here. Maybe this is a receptor on a membrane. This is the ligand coming in, and it's binding to the receptor and causing the receptor to transition to a more active state. So you could imagine, some mechanism. But mathematically, it's really simple. And the same goes for the others. What does increasing EP also increase E, which will decrease? Yes, there's a bit of to and fro here. So the question is, but doesn't increasing EP also increase E, which will decrease R? That is also happening. So there's a bit of back and forth here. So the question is, well, if I increase R, yeah, that'll increase E to EP, but then EP goes up and that'll form more E. That is true, but the net balance is that more E is made because there's more R. Yes, you will have an increase in the reverse rate, but it'll be smaller than the forward rate. So in the end, you will get more EP. All right. So there's a lot of balancing going on in these networks, and they can be sometimes tricky to understand what's going on, which is why we do computer simulation, of course. Okay. In fact, this is the explanation. Okay, so I'll leave you with this slide. You can look at it later. But this basically goes on what I just said, and I do it in language here, and I do it in a sort of symbol form here. Okay. So there's an initial increase in R, but then there's an even more of an increase in R because of the positive feedback. Okay. All right. So let's do an experiment. In fact, let me grab this. Oops. Actually, I need the model first, don't I? Let's grab them. Let's grab the model first. So let's get that. let's go to my, let's do a new one. R equals TE load a dot 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 dot, and paste that in. Let me run that to make sure there's no problem. Yeah, it looks like it's happy. Let me move that to make it tidier. So that's the model. Now let me go and grab this funny stuff here that's going on. I'll explain what this is doing. Okay, so I've got the model. Remember that S is the signal. I'm collecting two things. I'm collecting X and Y. X will contain the signal. Y will contain the output, and the output's going to be R, okay? R is the output. This is what I'm going to look at. So what I'm going to do is I'm going to have a range of S's of signal. I'm going to record the signal. Sorry, I'm going to assign the signal to S in the model. I'm going to run a simulation. I'm going to compute the steady state. And you may be wondering, why on earth am I doing that? Sometimes you have to help the steady state with a pre-simulation, and that's what I'm doing here, just to help it a little bit. And then I'm going to record S in X, and I'm going to record the steady state level in Y. And once that's done, I'm going to plot X versus Y. I'm going to do two things here, though. I'm going to use a marker, which is O. I'm using matplotlib here, okay? I'm using a marker of a circle. I'm just making this looking, just to make it look good. I'm making the interior color of the marker nothing, and I'm gonna make the edge of the marker red. Okay, so that's all I'm doing. So let's run this and see what happens. Once it runs, let's go. And there you go. Man, look at a strange thing that is. So remember that the x-axis is the signal, and the y-axis is R. So I think you'll agree that we're starting at low signal, we're starting at a low steady state, all right? And as I increase the signal, the low steady state seems to drift up. And then suddenly, I see a jump. Now that jump is the jump to the high steady state. And then as I increase the signal, that continues to increase as well, all right? So this is very characteristic of a bistable system. If you have an input, as you move the input up, you see this transition jump, okay? Now, I can also do, let me go back to the slides, and I'm going to grab some more code. Let me go to this other one. Oops. I'll grab this code. I'll put this in. What this one is going to do, it's going to go backwards. It's actually going to start at a high signal and go, and the signal, I'm going to decrease the signal. That's why I've got a minus here, all right? And I'm going to run it backwards from high signal back to low signal. So let's run this whole thing. It'll take a little bit to do this, probably. Obviously, Google's having a hard time today. Oh, there you go. Now, to differentiate between the two, I'm coloring the second one in blue, okay? Right, so I'm doing two experiments here. The first experiment, I'm moving the signal from low to high, and that's in red. In the second experiment, I'm moving the signal from high to low, and I'm doing this in blue. And so this is what you see here. So you see the red, that's the red there. We see the slow increase, then it jumps to the high one, keeps on going. But notice the blue one. The blue one is sort of hidden under the red. But you see the blue one starting high, it comes down, but notice it doesn't immediately jump down at the same place. It actually carries on, and it actually jumps down at a different place. All right. This is given a particular, this behavior has got a particular terminology to it. It's called hysteresis. All right. This is called hysteresis because we see that the transition happens at different points, whether I'm moving up or whether I'm moving down. All right. And let me go back to the slide, and I can show you this. I've combined the two. like this. There is an invisible line. Hold on, view, slideshow, here you go. There is actually an invisible set of dots in the middle here. They don't show up because those middle dots are unstable. Now I could write a script to grab those middle dots and plot them. But there are a set of middle steady state dots where that green line is, but they're unstable and I haven't actually managed to get to them, all right? But I probably could if I messed around a bit. I could find the unstable points along here and plot them as well, all right? Plot them in green or something. So this is your classic, what's called a classic hysteresis shape, all right? It's an S shape, right? That's called hysteresis. Okay. And there's a nice paper from James Farrell. He has a bunch of nice, very readable papers on this kind of topic. And he explains this hysteresis. But basically, you get transitions occur differently whether the trigger or the signal is moving up or moving down. Okay? All right? Now, there's one trick you can do. Do I have... Oh, I don't have the trick here. Ah, damn it. Let me see. I don't know if I can. Let me go back to the. I don't know if I can make this trick happen. This is going to be difficult. Let me change some parameters here. See what we get. I don't know if I can make this show you this trick. I should have. Otherwise, I'll show it to you tomorrow. Now, this goes back to the question. Yeah, that hasn't done it. This goes back to the question about long-term memory. Let me go back to the original one. Oh, haha, there's an interesting question on chat. Yes. Running time backwards. Let me go back to that in a minute, okay? That's a really good point. So I'm back. Have I? That must be has to be 0, does it? Was it 0, was it? We'll finish in a minute, okay? Should I show you this? There it goes. Okay, I'm back to that. There's some parameter in this model, I'll have to find out which one it is, where the parameter moves this entire curve to the left. It keeps the shape intact, but moves the curve to the left. If I move it sufficiently to the left, I can push this entire thing beyond the 0 point on the signal. So in fact, this piece here is moved to beyond the positive values of the x-axis, right? Now what that means is it's not possible for me to have a negative signal. There is no negative concentration. So I can never get to that piece here that's now been slid to the left. And what it means is there's no way, by moving the signal down, there's no way to get to the lower state without having to do a major disturbance of the system. And this may be one way in which differentiation works by making sure that the high state is in a place where it's impossible for the signal to reach the transition to the low state. I'll see if I can, I'll work on this afternoon and see if I can get this to work. But it's an interesting idea. And John Tyson has that in his paper, actually, where you can move the entire plot here to the left so that this entire section here from the left on is in the negative quadrant. And so it's inaccessible because you can't have negative concentrations. And all you're left with is this positive bit, all right? All right, so we leave it there for now. The next slide is on oscillators. And somebody has a very interesting point in the chat where they point out that this middle piece, how can I get this middle piece? There is a very clever trick you can do. And I've done it, it actually works. where what you do is you make every reaction rate negative. All right, make every reaction rate negative, and run the same simulation. In a sense, what you're doing, you're inverting the stability of the system. So the systems that are stable become unstable, and the systems that are the unstable states become stable. And one way to look at it as well, as the person on the chat says, is you're effectively reversing time. So when you run a simulation like this, you're actually running the simulation backwards. And so if you run the simulation backwards, all unstable trajectories now appear stable, and all stable trajectories now appear unstable. So if, and a quick way of doing that, because we don't let you do negative simulations in time. Oh, I don't think we do. You know, like my start is 50 and my end is 0. That will be
Yeah.
simulating backwards. I don't think we allow that.
We don't.
We do or don't?
No, we don't.
We don't. That's right. So you have to stick with the positive simulation time, but it turns out the way to mimic the simulation going back in time is simply to make all of these negative. That's all you have to do. Make every one of them negative. Make sure the entire thing is negative for all 4 of them. And if you do that, that's equivalent to running a simulation back in time. And then all the stable ones become unstable and the unstable ones become stable. And so it's a nice way of grabbing the green line. Of course, all these other lines now become unstable, so you won't be able to get them easily. But the green line now becomes stable, and you can get those points. Anyway, that's a good point to make. Okay, I think we've come to the end of the morning. Any other questions? Otherwise, tomorrow what I'll do is we'll start in the oscillations. I'll probably start it at 9 rather than 8 because Joe Hellerstein will, Joe Hellerstein will be doing model calibration, and then I'll continue from 9 on a little bit, and then John will do the.
So we're still meeting at 8. It's just oscillations won't happen at 8.
Yeah, we're still meeting at 8. Oscillations will happen at 9. Okay?
Same time, same schedule as
Yeah. All right. If there's no other questions, we can finish for the day. Okay. All right. Catch you tomorrow.
See you later.